So the main purpose of glycolysis is to break glucose down into two molecules of pyruvate while extracting some energy from it in the process. Now, I am going to go through all 10 reactions in glycolysis, but before we get into all that stuff, let's just start by splitting glycolysis right down the middle into a first half, the energy investment phase, and a second half, the energy harvesting phase. So you can see we start with glucose at the top, and what we do is we invest 2 ATP into it to make it less stable and easier to split in half. Now the way I like to think about this is I look at the fructose 1,6-bisphosphate, and I see the phosphate groups kind of as like handles, like you could grab them and just pull from either end and split it in half, which is pretty much what happens. So what you're left with is these two 3-carbon fragments, GAP and DAP, and they are processed through a series of reactions which pulls the energy out of them, and what you're left with is pyruvate. So that's the most broad strokes overview I can give you. You invest some ATP into glucose, destabilize it, split it in two, process the two fragments, and you get pyruvate. So step one, the first thing we want to do is get glucose into the cell and keep it there so that we can process it in glycolysis. You can see here on the left, I've got a glucose just hanging out in the extracellular space. And what it can do is it can go through this green hot dog looking like thing, which is a glucose transporter protein, and it can enter the cell. However, I've drawn a bidirectional arrow to indicate that glucose can go both into the cell through this and back out, and it's sort of an equilibrium. So that's a problem because we want glucose to stay in the cell. The way we solve this is through the first phosphorylation into glucose 6-phosphate, which is unable to pass back through the transporter. The enzyme that catalyzes this reaction is hexokinase, or in most tissues, but in liver it's glucokinase. I'm not going to go into too much of the enzyme stuff, names of them, for most of the reactions, but for the 3 regulated steps, I will. So since this one is an irreversible reaction, it's regulated, and the enzyme is hexokinase. So the second step is just a simple isomerization from glucose 6-phosphate into fructose 6-phosphate. The reason for this is just that fructose is easier to split than glucose. The other thing to note is that this is a reversible step, which is, again, a problem because we don't want the reaction going backwards, which is why we need step 3. Step 3 is another phosphorylation and a irreversible regulated step, which prevents the reaction from going backwards. So we go from fructose 6-phosphate to fructose 1,6-bisphosphate, which is really easy to say. Now you can see in the top right that we've gotten to this point where we've got our 6-carbon chain, we've got our 2 phosphate groups on there, the handles, and we're ready to pull this thing apart. The other thing too is that the enzyme that catalyzes this reaction is phosphofructokinase 1. So now we've split the molecule in step 4. We've gone from fructose 1,6-bisphosphate, and it splits asymmetrically into dihydroxyacetone phosphate, or DAP, and glyceraldehyde 3-phosphate, or GAP. Now the thing I want to but it's important, I'll tell you, is that GAP is able to go straight through the rest of the pathway, no problem. It goes right ahead to pyruvate. DAP, on the other hand, can't, and it needs to be isomerized into GAP in order for the reaction to proceed. So that's step 5, is the isomerization from DAP to GAP. Step 6 is a little bit confusing, I find, because it looks like we're investing more energy because another phosphate group is being attached here, right? We're going from GAP, we're adding in some NAD+ and a phosphate group, and we end up with 1,3-bisphosphoglycerate, or 1,3-BPG. I want to emphasize that this phosphate group is just coming from free phosphate floating around in the cell. It's not from ATP. There is no energy that is being invested into this. But what we're doing is somewhat similar to what we did in the first half of the pathway, which is what we're doing is we're attaching phosphate groups onto the molecule to destabilize it. But in this case, we're not going to be trying to break down the molecule any further. What we're going to do is we're going to make it so unstable that the phosphate groups want to leave and go and make ATP. So what happens here is an NAD+ comes in, the blue hydrogen, the top left, leaves, attaches to NAD+, you get NADH, and then the phosphate group comes in and replaces it. And you're left with 1,3-BPG. Step 7, as I said, we've got 1,3-BPG, which is like super unstable, and these phosphate groups want to bail. They want anything to just get out of there. So what happens? ADP comes in, the phosphate group goes, I'm out of here. It joins ADP to form ATP, and you're left with 3-phosphoglycerate. Now we've got 3-phosphoglycerate, but we've still got a phosphate group left on it, which is a problem, right? We need to get rid of that. So the next 2 steps are going to be doing a similar thing in a slightly different fashion, where what they're doing is making the molecule less stable again so that that phosphate group can leave. The first step is just a simple isomerization, moving the phosphate group from the 3rd carbon to the 2nd carbon to make 2-phosphoglycerate. Now that we've got 2-phosphoglycerate, what we're going to do is we're going to turn it into phosphoenol pyruvate, or PEP. If you remember your organic chemistry, you might remember that enols are super unstable, which is perfect, right? That's exactly what we want. We want this to be unstable. So what I've drawn in blue here is we've got our blue hydrogen and our blue hydroxyl group, and they're going to leave as water. What we're left with is phosphoenol pyruvate, super unstable, and as you might guess, in step 10, another ADP comes in, phosphate group goes, great, I'm out, joins ADP, you're left with ATP and pyruvate. And this is also the 3rd regulated step, which is catalyzed by pyruvate kinase. So that's pretty much glycolysis. I know that was a lot, so I'm just going to do a brief recap here. First half, all we're doing, 2 phosphorylations to get us from glucose to fructose 1,6-bisphosphate, make that molecule unstable. We split it into GAP and DAP. Now, the other reason I've drawn this is to really emphasize that the second half of the pathway happens twice because both of these 3-carbon fragments are going through it. So GAP goes straight ahead, you get an NADH out, 2 ATPs out, and you're left with pyruvate. DAP, isomerized to GAP, same process, an NADH out, 2 ATPs out, you're left with pyruvate. The net reaction is 1 glucose, 2 ATPs invested, but you get 2 NADH out and 2 ATP out because there's 4 there, but 2 of them are making up for the ATP you invested. And that's glycolysis.